Good morning, everybody. It's my pleasure to introduce Dr. Benjamin Belgrad. He earned his doctorate at the University of South Carolina, Columbia, and is now a senior marine scientist at Dauphin Island Sea Lab. Much of his work focuses on oyster restoration, oyster aquaculture and reef restoration, as well as exploring the role of chemical signaling in predator-prey interactions. And with that, I'll pass it off to him.
Thank you. Can you guys all hear me back there? Great. So thank you for having me here and being here today. As the title of my talk might imply, I'm particularly interested in individual differences, which is an intrinsic property of basically all populations where individuals can vary in physical characteristics that you see here. They can vary in their size, how fast they grow, how fast they move, their metabolic efficiency, all of which influence the performance of individuals, how well they can survive and go on to reproduce. Indeed, the composition of these characteristics in the population determines how well that population will be able to persist through time or if it goes extinct. And so individual differences are kind of an overarching theme of this talk. And two of the driving questions that we have here are how are phenotypic differences linked to the environment? So aside from the genetics, how is the local environment influencing individuals and their traits? And then what are the ecological consequences of these differences? And so one of the model organisms that I work with are oysters. So why do we care about oysters? It turns out that they provide a wide variety of ecosystem services. For instance, shoreline stabilization. Many estimate that shoreline stabilization is actually its most valuable ecosystem service by dollar value because it buffers against storms, prevents erosion, and guards that beachfront property that we all care about. Everyone wants to keep their condo from falling into the ocean. They filter our water. They also provide nursery ground and habitat for a wide variety of other species that we care about, both for economics and the ecology of the system. And finally, we eat them. These guys comprise a multi-million-dollar fishery each year. So number of ecosystem services that they provide. Unfortunately, oyster reefs are one of the most degraded marine habitats globally, where estimates say we've lost about 85% of our natural reefs. And so here at the top, there's a picture of a healthy reef compared to a low-quality reef. And you might notice that the low-quality reef right off the bat has a different color. There's more algae in the system. And all of it has turned from live oysters at the top to basically dead shell hash. So you lose a lot of that structural complexity that allows organisms to have a habitat or hide and grow. Where I work in Alabama, we've actually had to periodically close the entire fishery just simply because there are no longer enough oysters to harvest from there safely. And this is a nationwide, it's a global issue. This is data taken directly off of NOAA's database that you can find online right there, where it shows from 1950 to today that what we're harvesting today is a mere fraction of what we were harvesting 70 years ago. This is for the entire nation of the commercial catch rate of oysters. If you go back even further to the 1800s, the picture actually just looks even more grim to what we were used to able to harvest from the fisheries. So what can we do about this? Well, because oysters are so important and because they're so degraded, there's actually a massive global effort to return populations to what they once were. And so we're doing this in two primary ways. The first is through aquaculture, and the second is through a reef restoration. Now, aquaculture is where farmers will grow these oysters as isolated individuals, shown here in these baskets. And this is ideal for that premium half-shell market that you will find in the restaurant industry. They can sell them for a high price. Conversely, in reef restoration, what they do is they'll put out a hard substrate or a hard substrate seeded with babies, and this will create that more natural, complex reef structure that provides all those habitat characteristics that we care about. But these oysters are a little bit malformed, so it's best for the cannery industry. and they'll grow in clusters like so. Now, the difficulty with oyster aquaculture is that it is extremely labor intensive. So in order to do this, you have to grow them as isolated individuals. You're growing them in cages that are suspended off the seafloor, and this helps protect them against predators. But it takes a massive effort to prevent these cages from becoming completely biofoul. So farmers have to go out each week and de-biofoul them from all the epibionts that are growing on the cages. And so this does not have great economy of scale. If you want to grow more oysters, you need to buy more cages. You need to have more manpower to maintain these cages. We're working on ways to make this more efficient, but it's not there yet. It cannot supply the massive demand necessary for both the ecology and the economics that we used to have. Conversely, it's really easy to just toss out hard substrate or seed hard substrate and toss it out to grow into a natural reef for reef restoration. The issue here is when you go and toss these individuals out, they're tiny. And just like we love to eat oysters, everything else in the ocean loves to eat oysters too. So it's not unheard of and actually fairly common for people to lose 90% of what they put out there within the first couple of weeks of just planting baby oysters out there. So what can we do about this issue? Well, that's where my lab comes in, where we look at induced defenses, which is a widespread phenomenon where organisms in the presence of predators will alter their morphology to reduce how likely they'll be consumed. For instance, they may develop spines, a stronger exoskeletons, or a larger body to prevent the gape-limited predators from eating them. And it turns out that oysters do this too. In the presence of predators, they will actually grow a harder shell. Now, typical induction experiments look like this, which are laboratory-based. They're in closed systems, and they generally work on the order of a couple hundred individuals. And one of the issues with the closed system is that concentrate predator cues way beyond what you would naturally encounter in the environment. And so that makes it so that they'll experience more fear than they may normally do. If they look at survivorship, it's generally, again, laboratory based on short time scales. We're talking on the order of hours to days. And so what we wanted to determine is, can predator induction work on a commercial scale, where instead of inducing a couple hundred individuals, we're inducing tens of thousands to millions of individuals simultaneously. And in order to actually grow oysters naturally, hatcheries have to use a flow-through system where it's continuously pumping seawater in and then having it go out of the tanks, which really can dilute cues. So can it actually work under commercial settings? And the second, to what extent does predator induction actually enhance survival in the wild, where they actually are placing these oysters, particularly over longer time frames that we care about and that are relevant ecologically? So to get at these questions, we actually partnered with a commercial oyster hatchery, the Auburn University Shellfish Laboratory, and we grew what's affectionately called spat on shell, which this is one of our shells. And there's one of the baby oysters that they had seeded onto that. And there would literally be up to hundreds of baby oysters growing on a single shell. So when we have our flow-through system at this hatchery, shown here in our raceway tanks, we would have 4 tanks continuously running with about several thousand shells, each with dozens to hundreds of baby oysters grown onto them. So tens of thousands of baby oysters are in this system, and we would have 2 treatments. We would have caged blue crabs that would regularly be fed oysters to continuously provide predator cues, and then we would have our empty cages as controls. And then we would grow our oysters in this system for 1 and 2 months. And so the normal amount of time that a hatchery would be willing to nurse their oysters before they go and plant them onto the field is 1 month. But we want to also see, is there a significant advantage to growing them 2 months? Can we provide an economical reason for why hatcheries might want to nurse them even longer into the field and keep them in the hatchery system before planting them? So after they are grown like this, we would assess subsets of our oysters. We would measure the length and then measure how much force was required to actually crush a oyster using a fancy instrument called a penetrometer that measures the amount of newtons it takes to actually break the shell. So what did we find here? Well, to walk you through this, on our x-axis, we have oysters that are grown for 1 month and 2 months, and the dry weight of that oyster shell or valve. And for the purpose of this talk, green represents our control individuals not exposed to cues, and blue represents our induced individuals exposed to predator cues. Blue exposed to blue crabs is the way to remember that. And so after 1 month, there really wasn't a change in shell weight. After 2 months, the oysters definitely grew, and there started to be a significant difference in the shell weight. How about the shell strength? That's on this figure. So on our y-axis, we have what's called standardized crushing force. We've standardized it to size, since larger individuals naturally tend to have larger, stronger shells. So we compensate for that. And it's so at just 1 month of predator cues, induced oysters had about a 40% increase in shell strength, which is if you could suddenly went from being able to bench 100 pounds to 140 pounds if you are trying to equivalate that strength. Oysters that were grown for 2 months, that actually jumped even further, up to a 63% increase in the hardness and strength of those shells. But what's really fascinating here is when you compare across months, the induced oysters have roughly the same shell strength as normal oysters grown for 2 months. So essentially, induction added an extra month of shell strength to them when you grow them with the predator cues. But what we really care is just how this influences the survivorship. Does this actually help? Is it worthwhile? And so to do that, we composed a field experiment where we would take, again, subsets of our oyster shells and spend an enormous effort scraping them so we would have 10 individual oysters per shell, where we would mark our oysters with different colors depending on which treatment they were given. And then we would zip tie them to PVC poles in pairs. So there would be a control pair and an induced pair all along that. Some of the pairs would be caged to exclude predators so that we would know that mortality was from predators and not water quality per se. And then we'd go and plant them out into the field for our 1 month grown oysters and our 2 month grown oysters. And so we partnered with an oyster farm where they allowed us to go onto their property and just set these PVC poles out there for predators to munch on. So what did we find? Well, this is a survivorship curve where on our x-axis, we have the number of days they were out on the field. And on our y-axis, we have the proportion of individuals still alive, where the black bar is 1 month nursed, and then the blue bar is 2 months nursed. And so you get this very strong trend downwards that oysters were rapidly dying, that this is for the uncaged, that they were being consumed really quickly. So that's good. We want to actually see, is there mortality difference? Now, it might strike you as a little bit odd that the 2-month-old oysters actually have much higher mortality than the 1-month-old oysters, even though the 2-month-old oysters are supposedly bigger and stronger. Why is that? Well, because they were grown in the nursery for 2 months. That's a change in the season, and the environment changed over time. So in the spring, it's kind of fresh water from all the river inflow. And in the summer, it's actually saltier, which allows oyster drills to come and recruit onto these reefs. So oyster drills do not like fresh water. They love salt water, and they just went to town. So in the northern Gulf of Mexico, these snails are the bane of oyster farmers' existence. They are like a plague of locusts. They will be everywhere. And you can see that they were just clustered on the shells. They were even laying eggs on our shells as if. So it was great to see our oysters rapidly disappear like that, but it provided some good data for us. So what we cared about was just how much induction improves survivorship, which is this figure. So here, the solid lines represent our induced oysters, and the dashed lines represent our control non-induced oysters. And you might notice that the solid lines are always above the control oysters. So induction was always improving survivorship for us. And just how much is that? Well, if you actually to measure the percent increase in performance, so this is basically the survivorship of the induced oysters over the controls. And so this black line right here represents oysters grown for 1 month with predator cues. Over 30 days, the amount that they were increasing in survivorship over controls went to up to 50% increased survivorship. If you had exposed the oysters to blue crabs for 2 months, that percent increase in survivorship just grew and grew to nearly 300% increase in survivorship. So that's great that we are actually seeing some of the benefits of induction. But this experiment led to all sorts of other questions. One of the big ones was we were exposing our oysters to blue crabs, and they were being eaten by oyster drills. So do oyster defenses depend on the predator? Blue crabs normally consume oysters by actually crushing the shell of baby oysters, versus an oyster drill, like their name implies, they actually can burrow into the shell to eat oysters. So do they develop specific defenses depending on what type of predator they're encountering and how that predator feeds? What is the spatial variability in the benefits that they provide of inducing an oyster? And then does the benefits of induction vary by the predator regime that is encountered when you go and plant them at different locations? So we did a whole other experiment, again, using flow-through systems. This is another flow-through tank system at the Dauphin Island Sea Lab, where we had, again, about 12 tanks here. And we had, as we're doing these experiments, are continuously evolving and improving our methods. So no longer spat on shell. Now we have affectionately spat on tile, which allows us to more easily standardize the surface area that we're working with and just standardize the number of individuals. Again, we had thousands of baby oysters, and our 3 treatments that we had this time were caged blue crabs, caged oyster drills, and then a control of no predator cues added whatsoever. And we chose to grow them for 1 month because that's really what the farmers and the hatcheries said that they would be willing to nurse oysters for, even with our data. So what did we find? So this is where we planted our 7 sites across the Mobile Bay region. So this is the Dauphin Island. This is where I work right here. And so we had 3 sites here. This is where most of the oyster farms in Alabama are located. And then this one is kind of interesting to note. There's some farms here, and there's also a nice freshwater source right here that'll come into play later. We then were able to more easily mark our tiles depending on which treatment that they were given. And then we would zip tie them now to rebar poles in triads. So one of each of those 3 treatments added to poles, and we put out basically thousands of poles to monitor and check on a regular basis and count the number of individual oysters still alive. So what did we find? This is survivorship after just 1 week. Now this figure is a little complicated. Let me walk you through it. Again, blue is exposure to blue crabs. Red is now exposure to oyster drills, and green is control. On our x-axis, we have the different sites that we planted oysters out, and on our y-axis, we have the mean number of oysters still alive on each of those tiles. And so there's some patterns that you might first notice, and that is that survivorship overall at a site varied greatly. Some sites had really high survivorship, some sites did not. And the effect of induction also varied. At some sites, induction did not really have much of an effect. At other sites, induction would have a great effect, where the oyster drills seem to be performing a little bit better than the blue crab exposure. Conversely, at other sites, the blue crab exposure seemed to be doing a little bit better than the oyster drill exposure after just 1 week out to the field. And if you actually relate that to the location where we planted our oysters, we found that it kind of matched really closely to our predator regime, where oyster drills were the dominant predator. That's where they were, that exposure type was doing better. Where blue crabs were the dominant predator, and where there were less predators in general, that's where survivorship was higher and they were doing better. And then where there was just tremendous high densities of predators, that's where we had the highest amount of mortality. And so we were curious. This was just a week. We wanted to actually see how well this trend went through time. So we had two of our sites where we periodically went out even longer. And so that's this figure right here, where again, this is the performance increase over the controls, where at the site that was dominated by oyster drills and there was really high predation, survivorship by really increased when you induce them. Particularly with oyster drills, they were performing better than if they were exposed to blue crabs. Now you might notice that there's no more lines after here. And that's because after just about 2 weeks, all our oysters were eaten because there was just so high predation. At the other site, where there was lower predator density and it was dominated by blue crabs, again, we saw an increased performance when they were exposed to blue crabs, over 30% increase in survivorship, versus when they were exposed to oyster drills, that performance wasn't better at all. There's almost like there's maladaptation that's going on here. So you might want to match your predator cue with the type of environment that they're going out to, or at the very least, use blue crabs because that seemed to have increased survivorship regardless of the predator regime, which is what we've been doing since. So this leads to our key question here. Does induction actually increase survivorship at the restoration scale? So to do that, we actually partnered with the Nature Conservancy and the Mississippi Department of Marine Resources to build reefs and actually expose entire reefs worth of oysters to it, where we would have 9 metric tons of oyster shell seeded with 12 to 18 million oyster larvae, depending on the reef that we were building. And they were, again, exposed in 2 treatments, either caged blue crabs or no cage controls. Here's one of our cages where we'd have a blue crab. Here's another of our flow-through system that you saw earlier. That this time is completely full of multiple tons of oyster shell, and about 3 million babies would be in each of these tanks. And so we would then culture them for 1 month and go and construct our reef, where we had take these from a pickup truck to a boat and then shuttle them to our reef site. And so this is one of our intertidal reefs where we're laying the oyster shell down. And so this is our reef at low tide, where you can see the entire setup. And how we would arrange that is in columns, so that this would come from one control tank, this would come from an induced tank, a control tank, an induced tank. So we know exactly what treatment the oysters were exposed to when we would periodically go and resample the reef over an extended period. And so how did survivorship actually improve? Well, that's this figure here. After 1 year, which in the northern Gulf of Mexico, because it's so warm and phytoplankton is so abundant, is just how long it takes to grow oysters there. They're already ready to market size after just 1 year in Gulf. It's great. On our x-axis, we have our 2 treatments. And then on our y-axis, we have survivorship measured as relative spat per shell. Relative to what? Well, it's relative to the number of baby oysters we initially planted out there. So you can think of this similar to as a proportion or a percent of oysters still alive. And the key finding here is that after a year, induced oysters had a 68% increase in survivorship, which is massive. Now contrast that. You might also notice that these numbers are on the low end. So we're talking, for our control oysters, we had about 2 to 3% survive after a year, versus our induced, we were having about 4 to 5% survive after a year. But you have to remember that on the scale of commercial reef restoration and aquaculture and wild populations that are spawning millions to hundreds of millions to billions of individuals, that 2% total increase means that you are literally having hundreds of thousands more individuals survive to adulthood and market size, which at those numbers is the difference between a reef going extinct and being able to actually persist through time and become self-replenishing, just at those margin levels. So this is really promising for using predator cues to increase oyster restoration. However, induced defenses come with cost. You can't get something for nothing. It takes energy to actually build and increase that shell strength. And oysters and organisms in general have a finite amount of resources. So they generally have to reallocate resources from one thing to actually build these defenses. For example, with our tadpoles that I showed you earlier, these ones grew a lot bigger in the presence of predators to grow, but they no longer had legs versus our control. So they would delay their development so that they could get to that larger body size. And smaller body size is another very common cost to grow in defenses. And it turns out that our oysters exhibited this cost. This is a picture of some of our oysters when we were first doing this. The control are much larger than our induced. So we were curious, just what are the long-term costs of induction? We're only inducing them for a month, and then they're going out into the field for at least a year before that. So do those costs persist through time, or do they dissipate through time as the oysters grow? Are there decreased value for harvesters in the amount of soft tissue that they bring to market and that people can consume? Or does that change the amount that these oysters can actually reproduce and grow offspring? These were all important questions that we need to answer to convince the industry. So to do that, we did another experiment using single seed culture. Now we worked with single seed this time for a couple of reasons. The first, it's a lot easier to measure individual characteristics when you're dealing with individuals rather than clusters of oysters that are altering each other's growth. And the second reason is because we were noticing that single seed individuals actually seem to have greater costs of induction compared to spat on shell. And we're not 100% sure why, but our estimate is for a single seed oyster, it has to strengthen both valves to actually induce and grow. Versus a spat on shell, it has the substrate that it's settled on and has a freebie of one side of it. It doesn't have to induce or grow a shell on much. It just has to strengthen one of those shells. So by working with single seed, we're getting the most conservative estimate on how great these costs are. And so you might recognize this flow through system, but to grow single seed oysters, it takes a lot more labor. It's basically trying to make sure you don't get grains of sand sucked down your drain, and they'll actually float on the surface of the water if you don't break the water tension. And so there's a lot of little tricks to grow in single seed oysters that we had to learn. And then we would, again, grow literally thousands of these oysters that we were inducing, and then would plant on oyster farms to have them go through the normal commercial process of what it takes to grow oysters to market, where they'd be in these floating cages. These are our oysters. They would be tumbled regularly to get that cup shape that farmers like. And this is just our oysters from one of the farms that we were growing. So again, literally thousands of oysters that we were inducing. And then we would periodically take subsamples where we would use our penetrometer and we would dissect them to measure their soft tissue and their overall performance. So what did that look through time? So this was our shell strength. We wanted to make sure our oysters induced, and it turns out they did. They were about 25% stronger than controls. Now, how did that express itself in costs? Well, this was the shell size. So we have the length on the y-axis, and it turns out that they were about 17% smaller. And these were those oysters. And so you can actually see the difference, which is great. I love it in research when you don't even have to do any sort of data analysis or statistics. You can just look at the data and be like, oh yeah, that's definitely not the same. If you actually went and looked at the soft tissue, that cost was even greater. It was a 44% decrease in the amount of soft tissue that induced oysters had produced right after they were done being exposed to predator cubes. So how did that influence their growth over the time that they're getting to market? And so that's this figure right here, where we have number of days they're out on the farm and the soft tissue weight, which was the greatest cost that we were seeing for them, was in the soft tissue. And so for the first several weeks up to the first 3 months, there was a significant decrease in the amount of soft tissue that induced oysters had. But after about 7 months, there was no longer any difference between control and induce. And after about a year's worth of time, differences started to be in which cage they were held in, what were the local environmental conditions, rather than whether they were induced or not. Those costs completely disappeared. If we look at the amount of reproductive effort that our oysters were producing, that was measured as gonadosomatic index, which is a measure of the amount of reproductive tissue that is invested towards reproduction. And that was not difference between our controls or induced. In fact, our induced had a slight trend to have a little bit higher reproduction. Perhaps we scared them and made them want to get their gametes out quicker. So this is all promising for using predator cues to stimulate oysters. But what is the physiological mechanism behind this defense? What is going on here to increase that shell strength? To do that, we actually zoomed in onto the shell microproperties and partnered with the College of New Jersey to look at the fine characteristics of the shell, where we would take some of our samples and embed them in epoxy. And so you'd have a shell, and then you would cut it in half. And then you're seeing basically the side of the shell as on this figure. And then you can use an even more precise penetrometer shown here, which has a hard diamond tip to it, that if you slowly at a known rate and at a known amount of force embed it into your material, you can measure how big of an indentation that makes in the material. And by measuring the size of that indentation, you can get a measure of hardness. So this is a very common technique used in material science, particularly in the steel industry, to make sure that your materials are up to code. And you can do this with just about any material you care to name. And so we could actually measure the hardness, the micro hardness of the shell layers using this technique. So this is a panoramic shot under the microscope of one of our oyster shells to orient you here. This is the umbo, or the hinge of the oyster shell. This is the growing edge of the shell. And then this is where the body of the oyster would lie versus the environment. And if you were to zoom in on this, you can actually see the individual layers for the oyster shell. The harder outer prismatic layer and a much thicker, softer inner foliated layer. So we could actually go and periodically measure along transects to see just how thick the oyster shells were. So this is the layer thickness, and then the different colors represent the different layers here. And so it turns out that our induced were about 15% thicker shelled than our control, but that was not significantly different, partially because we only had 10 samples to work with, a low sample size due to the intensity of measuring these techniques. And there's a whole lot of individual variation here that can mask that. But how about if you look at the micro hardness of those layers? So if you do that, this is our foliated layer, and that is that diamond cross section that I was talking about earlier. So if you measure the lengths of those diagonals, you can calculate out what's called a Vickers hardness on our y-axis here. And so it turned out that our induced oysters had about a 4% harder foliated layer than our controls, and that was significant. If you look at the prismatic layer, that in general is a harder layer than the foliated layer. And that again was about 3% harder for induced oysters than control, and that was significant, which is really cool to see. That it just demonstrates that it just takes a small change in the material properties of a substance to have large effects on the overall strength of that substance, to have those cascading ecological effects of survivorship and costs of growing, just from this really small 4 to 3% change in the materials of the shell. Now, if you're curious, this is actually what those layers look under the scanning electron microscope at 10,000 magnifications. And so we're currently actually looking at even more fine details of the shell lengths and the actual prisms within the foliated and prismatic layer. Now, this is what's changing in the physiology. But what is inducing this shell change? What is the chemistry behind that? And to get at this question, we partnered with Georgia Institute of Technology. And so predator-prey interactions are really important for the ecology. They alter community composition, they mediate trophic cascades, they increase biodiversity, and they affect the distribution of other species. And particularly in aquatic environments, these interactions are heavily mediated by chemical cues. For example, as shown here on this slide, you can see these are basically hermit crabs that are being attracted to an injured conspecific, just the odor from it, because they want to go and find a new shell. They know that that individual has died and there's a shell available, so they'll swarm around. Here you have fruit flies tracking an odor plume. Here you have a bunch of fish tracking the plume from a bait. Animals rely on these chemical cues to know just where their prey are, how to track them, and prey rely on them to determine how risky their environment is and how dense the predator regime is and what predators are there. So chemical cues are really, really important ecologically. One of the things, though, is we know very little about what these molecules actually are that predators are releasing, that the prey are picking up on. And so that's what we sought to find out. So one of the challenges with actually identifying these molecules is that they are really dilute in seawater. We're talking just a couple parts per billion, which is too fine for analytical instruments to actually detect generally. Additionally, when you're looking at these chemical cues in the environment, it's this entire soup of literally thousands and thousands of molecules. Finding the particular molecules that are actually causing that response is like looking for the needle in the haystack. So how can we sift through that haystack and actually identify them? Well, to do that, we went straight to the source, where I went to a marine vet and asked her, hey, can you teach me how to catheter fish to extract urine? And she looked at me like I was nuts. So she said, well, let me think about it. And she went out to her colleagues and asked, hey, does anyone know how to do that? And they all looked at her like she was nuts. So we had to come up with this technique ourselves, where we learned how to actually anesthetize fish and then suture a catheter to the fish so that we could periodically extract urine from the fish on a daily basis using a syringe. And so that was surgically attached, and we could have several species of fish. We'd have black drum and sheep's head, where we would regularly collect urine from them. We'd also collect the mantle fluid from oyster drills. And then we also learned how to catheter blue crabs and stone crabs, which was actually a lot easier than doing it with the fish. That you could do by hand, but it's also different to do it from stone crabs and blue crabs. It's a lot harder with stone crabs because these guys are armored. And so doing this leads to some interesting headlines on what you might encounter. But what are we doing here? So after we would extract our urine from our different species, we would create what were master mixes, where each mix would be from that one particular species and have different individuals making each mix. So we would have 2 individuals of blue crabs make one mix, 3 individuals of blue crabs make another mix, and then we would aliquot these out and then put them in a -80 freezer so that we could continuously expose our oysters to the exact same chemical mixture and cues. And so this allowed us to rely on individual differences between predators to actually help us ID our molecules. How? Well, let me walk you through the magical world of metabolomics. So the theory behind this is you'll run a bioassay where you'll measure the response strength. It doesn't matter what really. For us, it was shell growth. It could be anything you choose to name. And you expose your individuals to different mixtures. And so dependent on how potent that mixture is of those predator cues, you will get a stronger response than a less potent mixture. And then you can do chemical analysis on that mixture. Usually you start with NMR spectroscopy, and that'll create something that looks like this, where you'll have a whole bunch of peaks at different amplitudes. And so each peak represents a different molecule, and the amplitude of that peak represents the concentration of that molecule. Now, because your mixture is from the same species, it will have the same molecules in it. So all of these peaks will align. But because your mixture is composed of different individuals that have differences in diet, differences in metabolic rate and how they process that material and digest it, you will have differences in the concentration of each of those molecules, represented by differences in the amplitude of those peaks. And so you can do some fairly complicated statistics in machine learning to actually cross-correlate the strength of your bioassay response to the peak amplitudes to identify which of those peaks and which of those molecular signals is actually responsible for your change in shell strength. And so for our bioassay, we would have jars of oyster and our spat on tile, and we'd have about 12 mixtures per predator cube for each species. And then we'd have about 100 plus jars per run that were constantly aerated. We would have to feed them multiple times per day for each jar. And we'd have to regularly add cues and change the water. Now, oysters do not like to grow in jars at all, but this is the ideal way to actually tightly control what molecules your oysters are regularly being exposed to. So what did we find? Well, this is the standardized crushing force of our oyster, how strong they grow. On our x-axis, we'll have our different treatments. And then these black dots represents the average shell strength, the performance of the oysters for that particular mixture within that treatment type. So we had different mixtures of seawater. For example, we also had different mixtures of a positive control of predator water. This was just water that blue crabs were naturally feeding oysters in, and we filled a quarter of the jar with this predator water. And they grew a lot stronger if they were exposed to that predator water. If we expose them to the urine of blue crabs, they also grew a lot stronger than if they were just given plain seawater. And notice here, so these two bars represents blue crabs that were either fed oysters or mud crabs before they were given their urine to the oysters. But notice how there's not much difference in the overall response, and a great amount of difference dependent on the mixture that they were given, which is perfect. A lot of times we don't like to see variation in our results. This is a time where we absolutely want to see variation in how strong of a response those mixtures gave. And so we were able to leverage this data to actually start to find the molecules. What was also really cool was as you increase the concentration of urine that the oysters were exposed to on the x-axis, you get what is basically a textbook dose response curve, where at very low concentrations, starting with our negative control, there wasn't a large increase in shell strength. As you increase the concentration, how strong those oysters grew rapidly jumped up to some threshold value where it didn't matter how much urine oysters were exposed to, they were not able to strengthen their shells anymore. Probably because it's a physiological limit to just how strong an oyster can get its shell over that amount of time. If we're looking at other species besides blue crabs, this was for our oyster drill bioassay, that produced an intermediate response. There was a tremendous amount of variation, but it wasn't as a whole stronger than the control. So we're looking into that still. How about our other species? This was, again, our negative seawater control, our positive predator water control. And then this was the urine from stone crabs, the urine from sheep's head, and the urine from black drum all produced significantly stronger oyster shells than the control. So this is really cool to see. First, it shows us that oysters can detect multiple different predator species in the environment and will respond to each of those predator species. And secondly, we now know that this is from this cue is in the urine of these organisms. It could have been from other goods. It could have been from the saliva. It could have been particulates that are sloughing off of their dermis. And so now we actually know where to go and dive down deeper. But we want to actually know what molecules are stimulating this response. So we'll actually go through NMR spectroscopy. And so each of these lines represents the spectral profile of one of our 12 urine mixtures for that particular blue crab assay. And so you'll notice a lot of peaks here, where some peaks were consistently really high for each of those mixtures, versus others. These peaks represent different molecules or molecule fragments, they vary depending on which mixture they were in. So again, we're seeing a lot of variation in the chemical profile of our mixtures. And so we found over 720 potential metabolites in the urine of just blue crabs alone. And through some machine learning models and genetic algorithms, we were actually able to narrow that down to 10 candidate molecules that we could go even further and test against. And so one of them was this little amino acid called trigonylene, which this is its characteristic signal right here. It was a very prominent peak. And so if we were actually go back to our bioassay and expose our oysters to just pure trigonylene, we'd find that that actually caused oysters to grow a much harder shell. Homarine was another molecule that we identified that will grow a stronger shell. And if you combine trigonylene and homarine together so that they're at the normal concentration you have in urine, that would also create a much stronger shell, a little bit better than if you just combine them alone, but not significantly different. So what can we take away from all of this? Well, that predator cues are a promising tool for restoration. They're a non-toxic mean to control pests. And it doesn't just have to be with oysters or bivalves. There's a whole slew of other organisms you can do this with. So I know that China, they have some researchers that are looking to use locust cues to help grow rice and make rice pest resistant. On the West Coast, kelp growing and restoring kelp is a big issue because you have urchins that are constantly eating kelp barrens and making those disappear. You can use starfish cues to actually scare away sea urchins so that your kelp can grow better. Basically, it's whatever organism you choose to market that you can probably find a predator cue to help control pests to some degree. Another key thing is that identifying these molecules opens up options. So you don't have to grow a predator into the system like you would if you have these molecules. I foresee a time where you could have bead bags of these molecules that you can synthesize in a lab and actually purchase online and just slowly drip and fertilize your oysters to grow stronger, similar to how we fertilize our crops with nitrogen to grow more robustly. Identifying these molecules also opens up options just for experiments in general. So a lot of prey responses to predator experiments often rely on putting individual predators into the system. And because you're working with individual predators, you are dealing with all the issues associated with individual variants in those predators and how scary they are. When you know the molecules and you know the cues, you can control just how much of a fear response you are providing to your prey species. So a lot better opportunities to standardize experiments across organisms and species. So I finally want to end that this is only the beginning of what we can do with induction and predator cues. It's really, the world is your oyster when it comes to just how far you can take this. And so I, of course, have plenty of people I would like to acknowledge, and I will take any questions.
Okay, so we have two students who are going to ask the first questions, Brianna and Farah.
Super awesome talk, very informative, super cool work here. So the question I had was, so those induced oysters are usually smaller starting out. Would it kind of affect the time that the baymen are able to, like the time frame that the baymen are able to put those oysters onto their cages? Because I know they have the spat and the small little juveniles, or before they get to a certain size, then they put them in the cages to grow during the season. Like, do you think that would affect the time frame that they're able to do that?
Yeah, so the question is, because induced oysters, when you're done with that nursery period, they're a little bit smaller than they normally would be for going out into the field. Does that increase the amount of time that you actually have to hold them in the nursery before they go out into the field? And potentially. It depends if the farmer is going to hold them in the nursery right up to the point where they can then go into a cage and then go out, or if the nursery will hold them beyond that size because of water quality issues, or because the pore size is just so small in the cage that they can go out even at that smaller size. And so my answer is it can, but it's really circumstantial, and it depends on just how much of a cost benefit that increases. If you have to hold an oyster for 3-5 more days before it goes out into the field, I would argue that's good. For reef restoration, they don't care about cage size at all. They just toss them out into the open ocean, regardless of what size they are. So for aquaculture and doing that single seed oyster, it might. But for reef restoration, not an issue, definitely. Yes.
I have many questions.
You get
It's
one.
a great talk, by the way. I will play the devil's advocate. Can the predation pressure be a factor of size, response to size, being smaller, so there is less predation on them? As simple as that.
Yes. So the question for those on Zoom is, because they're smaller, do they actually, are they less attractive to predators? So the predators go for the controls instead of the induced. And we've done some predator choice assays with oyster drills on this, actually. That's done by an RAU student. It's in review right now. It's a small paper, but it was our tentative first look at this, where we gave oyster drills a choice of, can they go to control, can they go to induce? And we had a whole bunch of assays to see, are they selected for that? And basically what we found is that the drills would not have a preference in which one they would go to, but they would fail more often in being able to consume that induced one. And so they had a much longer handle-in time for the oyster drill to actually break through and get into the shell than it was for the control. Could be dependent on the predator, though. So
yeah,
there's definitely more work to do on that, but tentative first look, nope.
I was wondering if, sorry, I'm like formulating what I was gonna say now. If you think you're like inducing a response that is happening at an earlier stage, just like already in the wild with like, oyster larvae that like spawn like in the wild, do you think they're already like inducing this predator response like when they're forming their shells? Does that make sense?
So are we causing them to strengthen their shell earlier than they would naturally? Is that the...
No, more of like is this like a naturally occurring thing that like happens like very, very early on before like we like in aquaculture are introducing the oysters into open water? And so like, is it kind of like a step that's already happening, do you think, in the wild that we're then having to bring in?
So
Because it's not happening.
yeah.
Captain.
So the question is, do oysters induce naturally in the wild? And because we are growing oysters in a nursery system that doesn't have predators, are we add in something that they would normally have if they were to grow naturally? I would say it's a little bit of both. So yes, oysters do induce naturally in the wild. But the concentration of predator cues that we provide in the hatchery is greater than they would normally experience in the wild, and it's more continuous than they would experience in the wild because the predators move about generally. And when we are measuring the oysters, the shell strength of oysters that are grown in the nursery, they do tend to be a little bit stronger than equivalent wild oysters of that same age. And that's really hard to measure too, because it's the oyster shell strength on a natural reef is incredibly variable. So we do think that it's a little bit beyond what they would get normally. And we also know that there is, when they start inducing is actually a little bit later in life. It's actually 2 weeks after they settle. And we have no idea what it's like for the larvae. That's another aspect that we're interested in. Other questions?
Hi, here on Long Island, we have an issue with an invasive species called spotted lanternfly.
Okay,
And I was wondering if you think that using predator cues could be effective for invasive species management.
I do think that it could potentially be, maybe not for the spotted lanternfly, but one of the things that I had in the real back of my mind is there's that Asian carp problem that's going across the nation. And if you could use predator cues to kind of scare carp away from water bodies that you don't want it to go into that river inlet, maybe. I think it just depends on your ingenuity that, yes, it can probably in some circumstances, but you're going to really need to tie it to your system. I'm not sure with the lantern fly because that's more aerial. You might be able to keep it away from crops. Like we have them. We're in my hometown of Ohio now. I'm
Yeah.
not sure if they are pests for crops. I think they're a pest for trees. So maybe if you have smoke of that cue around trees, possibly. But again, it depends on your imagination. Yes.
Do traumatized oysters taste?
That was my question.
That is a very good question. We would be happy for you to do taste tests, actually.
Volunteer.
So I might not be supposed to say this, but for some of our oysters, after we grew them out on the farm, we did munch on them because we had literally thousands of oysters left and didn't want to have them go to waste. So we did offer them with caveats saying, just be aware. No one got sick. And to my knowledge, no one could tell the difference in taste profile. Taste profile is very heavily day dependent in how salty it is. Yeah, we do know that fear responses do change the taste of meat in other organisms, but that's more of an immediate one. When oysters are grown out for over a year, my guess is no, because we don't see any of the other costs either. Yes.
Wondering if you did or plan to do any transcriptomic epigenomic work looking at some of the signals between the two groups, and then also if you looked at the offspring of the groups that you introduced versus those, you didn't see if there's any inheritance of those effects.
Yes, I have not done much genetic work at all. It's one of the things that I would love to do. Yeah, I would really love to do that sort of research. The closest that I've worked with genetics on that is comparing diploid and triploid oysters, because triploid oysters are prized by the aquaculture industry because they don't invest energy towards reproduction nearly as much. So they don't have as high of a glycemic index. They don't produce as much viable sperm. And because they aren't investing that energy towards reproduction, they tend to grow much faster. So I want to see if they have more energy for growth. Do they have more energy to induce? And it turns out that yes, they induce more, they have higher costs, but then at the end of the game, they grow out of those costs much quicker than diploid individuals. And at the end, it actually seemed to have primed them, where they induced ones actually grew even bigger than our control oysters by the end of a year. So there are definitely genetic components. The other closest way I've looked at that is through oyster spawns. And there is a high difference in the amount of induction that we get spawn to spawn. Some of that is through water quality differences. This year we had really poor induction because the water was a lot fresher, and so oysters aren't feeding as much. But we do see that some spawns perform much better than others. The amount of induction that we got could vary from about 25% to over 60%. So I am sure that there's a genetic component that we can tease apart if we so chose.
Or transgenerational.
Mhm.
Yeah.
Yes, or cross-generational to breed oysters to just naturally have a stronger shell. Breeding oysters is like breeding poodles. We breed them to grow stripes. Yes.
That's a great comment that you just mentioned, because I also wanted to know if those like induced traits were heritable. So like you could see that they were like that genetic changes or like.
So the answer to that is I don't know. This is purely environmental cause that we're seeing because we are doing this on the same offspring, same spawn, and just exposing them to the predator Q induces most, but we get individual variation in how much each oyster is inducing in the spawn. Part of that is, again, its local environment, but Q isn't evenly distributed. There's water currents and vortexes in there that can cause differences in Q exposure. But I would guess that there would be some genetic component that you could potentially breed into it. That you could get oysters to grow stronger and stronger, but we don't know. We haven't done that research yet. Most of the research into improving oysters right now is to make them more disease resistant. So Auburn University has just developed a strain of oysters that's more resistant to dermal. And they're making sure that there aren't any unintentional costs to having improved disease resistant oysters. No one's done that in induced defenses yet. Open field, yes.
A follow-up question regarding to the season. First of all, fantastic research.
Thanks.
And, I think there's so many continuing efforts on restoring our still fisheries and restorations in the Gulf of Mexico. I think part of my PhD research related to the pythonic oyster larvae.
Oh, nice.
Bioenergetic model with some big problems. So I think my question is related to like disease equivalents, like in some of your field experiments, how did you kind of isolate the fact the mortality sources from disease or its predation pressure? So, and
yeah.
apparently there's some seasonal variations. I guess the freshwater might not be an ideal environment for the disease, but the fall term might be a pretty good season for the oysters and good for the oyster larvae.
Yes.
Sometimes.
So essentially the question was, how do we know that this mortality difference is from predators and not, say, water quality or disease? And that's because for a while we would do these experiments with cage control, where we would have induced and control oysters in cages that excluded predators and controls that oysters were open buffet. And we found that if you cage them, they would survive, essentially. So long as you can guard against predators, you will have really good survivorship. If it had been disease or water quality that was influenced survivorship, we would have expected really low survivorship in those caged oysters too. And we did not see that. Oysters do fine often if you just protect them against predators. The problem with caging them is it's very labor intensive and high maintenance to make sure those cages don't biofoul. Plus, it's the material cost of having cages. And on the scale of reef restoration, it's not really feasible to cage an entire reef that's half a kilometer long and protect it against predators from entering that cage. You're not going to do that. So that's why we need to come up with other means to deter predators.
I think it's might be lunchtime. But Ben is gonna go to lunch. So I feel like anybody who has an outstanding question or wants to ask a second question, make sure you sit right next to him. Everybody sit right next to him. And thank you.